Hello, greetings to all of you, and to provide, I am Shadat. So today I would like to present and explain a bit about the article RON5 is critical for organization and function of the Toxoplasma moving junction complex. So firstly we go to the introduction. Apicoplasm evasion is highly coordinated process of attachment and penetration that relies on protein secretion events from two separate organisms, which are the macronyms and RON3s. Molecular enhancings are released onto the parasite's plasma membrane by secretion from the macronyms, allowing it to connect to the whole cell surface. The parasite apex is orientated toward the host cells after first attachment, and the RON3 contents are discharged. RON3 secretion is associated with the creation of the moving junction, which is MJ, a ring-shaped tight junction contact between parasite and host plasma membranes through which the parasite enters the whole cell. During evasion, a complex of the RON3 nets proteins RON2, 4, 5, and 8 localized to the MJ, providing a stable anchoring point for whole cell penetration, perhaps by contact with the whole cell cytoskeleton, as host cytoskeletal components relocate to the MJ and are necessary for evasion. The MJ also contains a molecular filter that prevents host plasma membrane proteins from entering the nascent parasitophorous vacuole, which is PV, making it non-fusogenic and shielding the parasite from lysosomal degradation, which is a role that the MJ-RON complex may fulfill. So we go to the very most important for this presentation, which is the result and discussion. So first, the study about RON5 is critical for evasion and vacuole formation but not ingress. So the only RON3 proteins known to localize to the moving junction are RON2, 4, 5, and 8, which are thought to play a key role in whole cell evasion. To test the importance of RON5 in whole cell entry, evasion assays were performed on RON5 CKD parasite depleted of RON5. In the absence of ATC treatment, these parasites have a slight decrease in evasive capacity compared to the parietal line, which is likely due to the decreased concentration of RON5 generated in the knockdown strain. Depletion of RON5 causes a significant block in evasion, demonstrating that RON5 is required for this process. The low level of evasion seen after ATC treatment could be due to the remaining RON5 level in some parasite, or it could imply that parasite lacking RON5 can evade, but only at extremely low level. Next, plaque assays were performed to better assess the evasion defect over the course of several lytic cycles. While wild-type parasite develop plaques readily in the presence of absence of drug treatment, the RON5 CKD parasites did not produce plaque in the presence of ATC, despite having a 100-fold higher parasite load. This finding demonstrates the important role of RON5 in Toxoplasma evasion and imply that RON5 may be required for the process. To find whether the parasite depleted of RON5 either exhibit an attachment defect or, more likely, initially attached normally but then detached following a failure to invade, as previously observed with disruption of RON8 and with knockdown of TGDHC7 or TGARO. So by inhibiting actin polymerization and impairing gliadin motility, cytochalasin D was used to stop the evasion process shortly after apical reorientation but before penetration. Parasite depleted of RON5 were shown to attach to whole cells with the same effectiveness as untreated or parietal line parasites under similar conditions. This evidence demonstrates that initial attachment is unaffected in the absence of RON5, and the failure to invade in the absence of RON5 is followed by gliding motility-based suppression, with parasite being washed away during invasion assay processing. Next, egress was induced using the calcium ionophore A23187 to determine the role of RON5 in this process. There was no difference in whole cell evacuation under this condition, as parasite with or without RON5 egress with the same efficiency. Overall, these findings show that RON5 is required for whole cell invasion but is not required for egress. Next, the MJ-RON complex is disrupted in the absence of RON5. So, the remaining MJ-RON components were studied by Western blot analysis to identify the effect of the loss of RON5 on the rest of the MJ complex. So in this study, interestingly, when RON5 is knocked out, the RON2 signal is also lost, implying that RON5 is required for RON2 stability. So next, to look on RON5C is required to stabilize RON2. To see if pro-RON5 is necessary for RON5 targeting, and if frame deletion was made in the HA flag double-tag RON5 expression cassette, deleting the region between the signal peptide and the suspected pro-cleavage site. This protein was found to be grossly mistargeted using both HA and flag epitopes, while this data implies that pro-RON5 is required for RON5 targeting. C-terminal truncation analysis reveals that pro-RON5 is insufficient. These findings show that the pro-region and C-terminal portion of RON5N play a role in appropriate targeting, potentially by guaranteeing appropriate RON5 folding and or enabling interaction with other MJ-RON complex members.